So I'm really fortunate enough to have a bit of time with you guys today to discuss one of my big passions, and that's how we can improve the outcomes of cancer patients. And we have a really big problem on our hands, actually, and that's the fact that of those that are online today, approximately 50% of us over the course of our lifetime are likely to develop one form of cancer or other over our lifespan. This is terrible statistics, and actually could be a whole new talk and topic of conversation. But what I want to really focus on today is, in my mind, the even worse situation is the fact that of those that are diagnosed with cancer, 50% are not likely to survive that disease and will die prematurely because of it. And there are numerous reasons why that is the case. A big driving factor of this mortality is the fact that we are diagnosing cancer in many indications at a very late stage. So again, this is for imaging scientists here on the call. This is an FDG PET scan, and you can see widely metastatic disease. And you can't cure cancer using surgical methods when it's diagnosed at stage 4. But we have a great arsenal of therapeutics to employ when we have disseminated disease, ranging from these targeted therapies, given the fact that we can now sequence the patient's tumor. We have, of course, immunotherapies that in some indications has led to curative outcomes. And then I think from my side, at least, the next level of cancer therapeutics, which is these targeted radionuclide therapies, which I'm going to touch upon a little bit in the course of my talk today. So the big problem is that despite all of these great therapeutics, resistance is one of the driving causes of mortality. And that's because tumors themselves are highly adaptive. So what we see here is, we have the initiation event that leads to this growth of the tumor, and tumor initiation could take many decades before we start to overcome the innate and adaptive immune system, and we get tumor outgrowth. So we get this initial tumor growth, and hopefully we're diagnosed at an early stage and we can choose the right therapeutic for the right patient, and then we get some tumor regression. However, unfortunately, due to the adaptive nature of these cancers, we have either innate or adaptive changes that occur, which lead to the increase of biomarkers that start suppressing the effects of these therapies, and we start getting resistance to them. And then we get regrowth and relapse of the cancer. And there are multiple biomarkers that are present that are causal for this drug resistance. And I actually think that imaging can help the problem of therapy resistance. And we can target it in two ways, in my mind. We can have a look at a very early stage after the initiation of these standard of care therapies to see whether the therapies are working or not. But maybe where we need to be going is trying to understand therapy resistance in far more detail. And I think imaging also plays a very important role where we start to see relapse. And this is going to be the focus of my talk today. So we really, within my group at King's College London, want to try and target this and improve outcomes for patients. We do this using a widespread approach, which is highly multidisciplinary. We have expert chemists that try and develop new imaging tools to try and identify therapy resistance in cancer. We have a range of different biologists and biochemists that use those tools to try and better understand response and resistance to therapy and the causal mechanisms of that resistance. But ultimately, we are using radionuclide-based imaging agents, and so we want to try and convert those into clinically applicable tools. So we need to trial those in patients ultimately. And one of the approaches that we've taken to try and better understand therapy resistance in cancer is to describe and to image this redox imbalance that occurs following therapy resistance. And by redox imbalance, I mean, as a simple biologist, effectively the numerator and the denominator between the harmful reactive oxygen species that are caused either innately or following therapy, and the way that these tumors respond to that harmful insult by protecting themselves using antioxidants. And as I mentioned, there's a range of endogenous and exogenous forms of oxidative stress that occur within these tumor cells. It could be either directly from radiotherapy, indirectly via chemotherapy, or just the fact that these tumors themselves are highly proliferative, leading to changes in mitochondrial metabolism, etc. And in these resistant tumors, they have an arsenal of protective mechanisms to try and stop the harmful causes of oxidative stress through effectively the efflux of these drugs, the production of antioxidants, etc. And within my group, we want to try and better understand this imbalance that happens in drug-resistant cancer. Many groups have taken the approach of trying to image the harmful reactive oxygen species that are produced following treatment. I'm going to use an analogy of why I don't think that's necessarily the right way to do it. Instead, within my group, we're looking at the antioxidants that occur subsequent to that oxidative insult. And the analogy that I like to use is that the reactive oxygen species, they're produced at such small timescales that you can't actually really see them. It's like dropping a stone in a very calm lake. You can't actually see the stone itself, but what you can see is the ripples that emanate out from that stone, from that reactive oxygen species, which are the ways that these drug-resistant tumors respond to that very therapy. And those exist on a timescale, excuse me, that actually is imageable. Okay, so today I'm going to talk about two approaches that we've made within this general scheme. First, I'm going to discuss the cysteine glutamate transporter known as XCT. I'm going to describe a way in which we can image the cancer stem cell marker aldehyde dehydrogenase as a surrogate marker of therapy resistance. And then I think a lot more excitingly is how can we convert these diagnostics to therapeutics? So we can suddenly switch a patient that doesn't have any therapeutic options to one that actually now we can therapeutically exploit. Okay, so let's start with XCT. What is XCT? Well, XCT I've been working on for over a decade. I love this amino acid transporter, but its function under normal physiological conditions is to take the dimeric cysteine, known as cysteine, into the cell in exchange for one molecule of glutamate. Okay, so it's very simple. Why is it important under normal physiological conditions, and why is it important in therapy-resistant cancer? Well, it's important because cysteine is the predominant form of cysteine in the bloodstream, so that's the way that cells utilize cysteine uptake. When it's taken into the cell, though, it's rapidly reduced to cysteine itself, and cysteine is the rate-limiting precursor of glutathione. And I'm sure many on the call will know that glutathione is a very important antioxidant. It's present in millimolar concentrations. It's actually the body's primary antioxidant. And in therapy-resistant tumors, glutathione is present in many orders of magnitude higher than healthy tissues and indeed therapy-sensitive tumor cells. And we can actually image the activity of this transporter XCT using a small molecule glutamate analog known as FSPG, tagged with fluorine 18. And FSPG effectively acts as another substrate for the transporter. It can go in and out, and that gives us a surrogate of its activity. Like I've said, we've been really interested in this for the last sort of decade or so. But prior to that, FSPG itself actually was developed by an amazing team that are now under the company name of Life Molecular Imaging. And they, with many other collaborators, used FSPG effectively as a competitor for FDG for tumor imaging, for a simple diagnostic agent. And you can see they trialed it in a range of different indications, and it works really nicely. It's rapidly extracted from the blood. It's renally eliminated, which for PET tracers is advantageous because you can just pee out the unbound radioactivity. And the only healthy tissue that takes it up is the pancreas, which is shown here. And hopefully you can clearly see with all of these different arrows spotted across these different cancer types, you get really high contrast to background images with FSPG. But that was not always the case. So in some patients, we got really high uptake, as I'm demonstrating here, but in others, it was less high. And that sort of killed FSPG for live molecular imaging as a general diagnostic. But I thought, as a biochemist myself, there is something far more interesting going on here. There is a reason why some tumors have low FSPG uptake. And that is actually not because the transporter expression changes. XCT is expressed at very high levels in nearly all of these different cancers, regardless of whether the uptake of FSPG is high or low. And so we did some really simple experiments. Given the fact that XCT links to glutathione, what we did is we oxidatively stressed some cells grown in culture. We did this using a range of different drugs, sort of shown here, with various different mechanisms of action. Some we got very high levels of oxidative stress, which is the x-axis, and in others we have sort of less levels of oxidative stress. And then we compared the levels of oxidative stress that these cells experienced to the magnitude of FSPG uptake, as shown on the y-axis. And hopefully what you can see here is we get really beautiful correlation between the extent of oxidative stress caused by these in these cells versus a reduction of this FSPG uptake. And of course, we took this forward into animal studies. This is a tumor in a treatment-naive mouse. This is an ovarian cancer xenograph, which lights up beautifully with FSPG. The black and white, for those that are not familiar, is just a simple CT scan. The color is FSPG. The more FSPG taken up, the hotter the signal. The colder the signal means less FSPG is taken up. And we can treat this exact same animal with doxorubicin, which is a standard chemotherapy used in the clinic, induces high levels of oxidative stress. And hopefully you can see this animal just 24 hours later has substantially less FSPG retention. So we can look at an early response to chemotherapy using FSPG. And this is maintained up to day 6. We don't actually get any difference in tumor size until day 6. So we're seeing differences via imaging far sooner than you would using the normal RECIST criteria. And FDG doesn't decrease until day 6 as well. So we get an early response. And of course, given the fact that this is PET, we can quantify this. So we get basically a halving of our signal 24 hours post-treatment, which is retained up to day 6. And again, to cut a very long story short, we looked mechanistically at why this was happening using metabolomics and cystine tracing. And effectively, as these tumors respond to treatment, they take in more cystine to create more glutathione. And because it's a bidirectional transporter, FSPG is effluxed at a higher rate. So we get less signal because we get more efflux of our FSPG from these tumor cells. So we've looked at various different treatments and we see the same thing. So this is just a more recent example when amazing PhD student of mine, Trish, treated, again, this is head and neck cancer with external beam radiotherapy. We fractionated the dose, so you can see here with the arrows what the fractionated dose looks like and when we did it. And then we look longitudinally at FSPG imaging over time to see if we can look at response to treatment. And what we did is we imaged here at day 7, pre and at day 7. You can see in the pre-treatment images that these tumors light up beautifully, and at day 7, we hardly get any signal in the tumor. This is the pancreas that is far more spread in the animal versus the human. And again, we can beautifully quantify this. So in the control vehicle treated animals, we get pretty high uptake that doesn't really change from baseline, whereas we get this 50% reduction at day 7. before actually we see any statistically significant differences in tumor size. So this actually can predict how those tumors are going to respond subsequently. I'm not going to go into too much detail. Mechanistically, it's super interesting. Again, it could be another hour-long talk, but we've also used FSBG as an early marker of immune checkpoint blockade efficacy. We see, again, there is this reduction following IO treatment at just 3 days. This is actually prior to extensive T-cell infiltration. Again, quite interesting here, and that's maintained up to 7 days. And this has been done in partnership with GlaxoSmithKline. Okay, so we can use FSBG as an early marker of treatment response, but wouldn't it be far better if we could actually predict whether these tumors are going to respond or not, rather than just have a try and see based approach? And that's what we've done looking at lung cancer. Now, lung cancer is the biggest cancer killer in Europe. It has a terrible prognosis of 5% survival after just 10 years. So things really need to be done better. Again, it's one of these indications where typically patients are diagnosed late and they present with therapy-resistant disease. And one of the reasons why they have innate therapy resistance is because in about a third of these patients, they have a mutation in this transcription factor known as NRF2, or its negative regulator KEAP1. Now, don't worry, I'm going to tell you how NRF2 and KEAP1 work, because you're probably not familiar with those, but just bear these two words in mind as I'm going forward. So about a third of these patients have this key mutation in one or other of these proteins. And what happens is those that present with mutation in either NRF2 or KEAP1 have a terrible survival rate in comparison to those that have wild type KEAP1 or NRF2. And the reason why a mutation in NRF2 is so damaging is because NRF2 is known as the master regulator of the antioxidant program. Again, remember back to earlier stages where I was saying like we're really interested in imaging antioxidants. There's more antioxidants. If there's more antioxidants present, these tumors are more likely to survive their therapy. Well, if we have this mutation in NRF2, we get hyperactivation in it. So it doesn't turn it off, it actually turns it on. And so we get numerous antioxidants being upregulated because it's a transcription factor. It directly drives the gene expression of multiple factors over 200 cytotoxic, cytoprotective agents, one of which, if I'm sort of highlighting it here just for cheating, means is XCT. The way that NRF2 works is under, again, normal conditions, NRF2 is transcribed and then it's quickly degraded by its negative regulator KEAP1. So KEAP1 binds to NRF2, it signals it for proteasomal degradation, and we don't have very high levels of NRF2. So NRF2 can't then translocate to the nucleus and turn on these 200 cytoprotective genes. However, when we get oxidative stress, or if we have one of these mutations, either in KEAP1 or in NRF2, we don't get very effective binding between the negative regulator and NRF2 itself. We don't get proteasomal degradation. We get a buildup of NRF2 inside the cytosol, and then it translocates to the nucleus where it turns on these genes. So this is it binds to the antioxidant response element here and turns on. And that leads to therapy resistance. Okay, so we asked the very simple question whether we can actually image NRF2 mutations in lung cancer. And we started by taking a panel of 8 cell lines, 4 that are NRF2 negative, so they have wild type NRF2, and 4 that are positive and have mutations in either KEAP1 or NRF2 itself. So you can see here by a simple Western blot, no protein of NRF2 present, expressed. And then we looked at XCT. XCT is directly regulated by NRF2, and as you would expect, we get a really good correspondence between the levels of NRF2 and the levels of XCT. Of course, because NRF2 is cytoprotective, we get increased numbers of antioxidants, glutathione being one. So if in blue, you can see all of those that are NRF2 wild type versus those that are NRF2 mutant, and you get at least a doubling of total glutathione inside these tumors, because glutathione ends up neutralizing the reactive oxygen species that are inherently present, we get a consequent reduction in these reactive oxygen species. So we just asked a simple question, given the fact that we have increased XCT transporter levels, do we see increased FSPG retention inside these tumors? Well, indeed we do. In a similar way in which we get increased glutathione production, we get increased FSPG, and actually the correlation between the levels of glutathione inside these tumors and FSPG retention is wonderfully correlated. Okay, so at the moment we're talking about correlations, but can we actually say that there's a causal link between NRF2 and FSPG retention? To do that, we took one of these tumor non-small cell lung cancer lines, A549, which has very high levels basally of NRF2. That's because it's got a mutation in KEAP1. We knocked it down, and then we restored the expression of NRF2. And what we saw again is we saw this very nice sort of matching of XCT expression. What happens with FSPG? Well, when we knock down NRF2, we get a subsequent reduction in FSPG, which is restored following the restoration of the gene itself. We can also take inherently low levels of NRF2, so this is NRF2 wild type non-small cell lung cancer. We can overexpress, albeit not so brilliantly, the mutated version of NRF2, so it sort of ends up accumulating in the cell. We get a little bit of an increase if you squint of XCT, and again we see a small increase in FSPG retention. I don't want to really harbor this point a little like too much, but again, we can use pharmacological methods to increase NRF2. We can use an inhibitor of the NRF2 KEAP1 complex, which effectively stops KEAP1 tagging NRF2 for proteasomal degradation. In NRF2 low cells, we don't really have any NRF2. When we add this inhibitor, we get an increase in NRF2 levels. Again, the same with XCT. We've done this for a range, so we get this increase, low to high, low to high, low to high with XCT. And again, we see the same pattern with FSPG retention inside these tumor cells when we treat them with this inhibitor Chi696. Okay, so what? Right, we're looking at it in cells. What we really want to do with PET is to look at it non-invasively in vivo, and that's exactly what we did. We grew NRF2 wild type tumors in the lungs of mice, and we imaged with FSPG. You can see this in this large tumor, we have relatively low uptake of FSPG, some uptake in the pancreas and also the kidneys and bladder. And then we can take the NRF2 mutant line, grow it up again in a slightly smaller tumor, and hopefully you can see there's quite a big differential. This is just showing you in terms of the video that this is actually in the lungs. This is not a subcutaneous tumor itself. And that took us a bit of time to get set up here at King's. Of course, we can quantify that. We see at least a doubling of signal in those NRF2 mutants versus the NRF2 wild type. And again, ex vivo, we can see that in the NRF2 mutant tumors, we have high NRF2, high XCT, and we can image that. We didn't stop there. We were lucky enough to collaborate with TracerX, which is a UK consortium looking at heterogeneity in lung cancer. And they have developed a number of patient-derived xenografts, which we were lucky enough to utilize in this project. So you can hopefully see the heterogeneity within these patient-derived tissue. This goes straight from the patient into the animal, doesn't ever see plastic. So you maintain some of that tumor microenvironment. In the NRF2 wild type, we see low levels of FSPG uptake, albeit some heterogeneity, as I mentioned. And again, in the NRF2 mutant tumors, these PDXs growing on the back of the mice, we see a nice differential, which again, we can quantify and look at ex vivo in a similar way. Using autoradiography with small tissue slices after we sacrifice the animals, again, really nicely we can pick up some of that heterogeneity in these PDXs versus what we would see with the normal cell line based models. But hopefully you can all see the magnitude changes dramatically between NRF2 wild type and NRF2 mutant. We've also gone the next stage working with our collaborators over at Moffitt Cancer Center in the US, where they've developed a beautiful mouse model of NRF2 mutation. These, following viral Cree administration, these genetically engineered mice spontaneously form lung tumors. This is because p53 it becomes knocked out by the virus and KRAS is activated. But we've also got these animals which have then a mutation in NRF2 or they're kept as wild type. This is the wild type version, so you can see multiple lesions within the lungs of these mice by FSPG, but look at the difference when we effectively hyperactivate the NRF2 pathway. It really lights up like a Christmas tree in each and every one of those lungs. And actually for a small molecule, we got something quite remarkable, which is up to 80% injected dose per gram of FSPG in some of these lesions. Again, we see a little bit of heterogeneity, which seems to link to the underlying levels of NRF2 activation in this genetically engineered mouse model. So we looked at this again ex vivo, and we looked at staining for XCT in this case. And again, see, we basically get a doubling of XCT in this genetically engineered mouse model. Okay, I'm going to just switch tack a little bit and talk about another marker, which is aldehyde dehydrogenase 1A1. This is known as a classical stem cell marker. It's also an antioxidant. So again, keeping with that same theme. The way that aldehyde dehydrogenase 1A1 works is effectively to neutralize the highly reactive aldehydes that are produced following toxic insult to the plasma membrane. So following, for example, chemotherapy or external beam radiotherapy treatment, you get membrane damage. This is shown in the form of lipid peroxidation. Lipid peroxidation leads to the production of multiple reactive aldehydes and a reactive aldehyde cascade. If left unchecked, these reactive aldehydes lead to DNA damage, which then can lead to further levels of reactive oxygen species, further lipid peroxidation, and ultimately it will push it forward and induce cell death. However, in the presence of aldehyde dehydrogenase 1A1 at high levels, these highly reactive aldehydes are neutralized to their more benign carboxylic acids. This means you don't get this cycle of DNA damage, lipid peroxidation, and subsequent cell death. So aldehyde dehydrogenase 1A1 is actually known to be a causal factor for therapy resistance. So yeah, this is just sort of highlighting what I've just described. So we were really interested in trying to see if we could image aldehyde dehydrogenase 1A1, ALDH1A1. And we did this using a substrate-based approach. So we effectively had a radioactivity attached to one of these aldehydes that could passively diffuse in and out of the cell membrane. And in the presence of aldehyde dehydrogenase, the aldehyde was converted to the carboxylic acid. The carboxylic acid is negatively charged, so it can't wash out. So effectively, you just get an increase in signal inside these tumors, which have very high levels of ALDH. Where ALDH is not expressed to high levels, we get washout, we get washout here, and so no signal. And this was the focus of many years of work from Raoul and Rene, two excellent postdocs in the group. Okay, for the chemists that are out there, I'm going to show you some structures. We're going to go through this quite quickly because of time, but yeah, I kind of wanted to go through like how we got to this stage. So what we did is we took this gold standard inhibitor of aldehyde dehydrogenase, known as DEAB. And what we did is we changed the electronics along the ring to convert it from an inhibitor to a substrate through the addition of this carbon. It actually works as a very good substrate, but it's not a very good radiotracer. So we needed to connect it to a different backbone. So we did some medchem around this to result in our final small molecule radiotracer, which effectively looks a little bit like DAB here in red, and it has this reactive aldehyde. And actually this worked beautifully in cells that have high levels of aldehyde dehydrogenase. This is an HPLC chromatogram. The retention time of our sort of substrate here is later on, shown in red. So here where we don't actually have any levels of high levels of aldehyde dehydrogenase, all we see is a little bit of this parent, effectively substrate. not really much conversion to anything else. However, if we incubate the same radiotracer in cells which have high levels of aldehyde dehydrogenase, the substrate is completely gone. Instead, what you get is you get the product, which is the carboxylic acid version. So actually, intracellularly, it's not only trapped, but it's retained as this carboxylate. However, this was all well and good, but we're using a highly reactive aldehyde to drive this mechanism. And unfortunately, when injected into the bloodstream, this is rapidly oxidized. So it actually doesn't work very well as an in vivo imaging agent. It works perfectly in cells, but not really anywhere else. So we took a different approach here where we wanted to try and protect this highly reactive aldehyde with this protecting group. So we effectively had a pro-drug radiotracer. And this protecting group is liberated by esterases. So we thought that we could deliver this protected group and the intracellular esterases will cleave, leaving the reactive aldehyde, which then could get trapped if you had high levels of aldehyde dehydrogenase. Again, works great in theory. In cells, actually, it works beautifully. So we can take these therapy sensitive cells, SCOV3IP1s, and then their resistant counterparts. So this is formed actually from exactly the same cells. Intracellularly, we get a doubling of the radioactivity. So it's cleaved, and then in the presence of aldehyde dehydrogenase, it's only in these drug-resistant cells we get good retention. If we inhibit aldehyde dehydrogenases with that DAB, as I mentioned earlier, we get really no retention inside these cells. This is just shown by an intracellular chromatogram of looking what happens with the radioactivity. This is our protected group here, which we see in high ALDH expressing cells. This protecting group is cleaved to the reactive aldehyde, and the reactive aldehyde is then converted to the carboxylic acid. If we inhibit aldehyde dehydrogenase, we still get that esterase-mediated conversion to the reactive aldehyde, but we don't get the conversion of the reactive aldehyde to the carboxylic acid. So the theory works. However, in vivo, it was rubbish. No matter what we did, if we changed the molar activities from low to high, so that's basically adding in cold compound to try and block it, we didn't get any differential between the therapy sensitive and the therapy resistant tumors here and indeed here. And the reason for that is that effectively, although in cells, we ended up protecting this rapid oxidation in the blood, again, very quickly we have esterases that are present in the blood that converts the protected radiotracer and takes it all the way through again into this carboxylate. So we were never actually delivering this aldehyde substrate to the tumors. It was just converted as the negatively charged carboxylate, and so nothing ever really got in. But it was good in theory. But what we wanted to do is very quickly change tack. So what we did is we created a new generation of radiotracers that's based on something completely different. This time we wanted to use a well-known inhibitor of aldehyde dehydrogenase and create a radiotracer. And we did this by using sort of copper catalyzed radiofluorination and radioiodination. So we created a hot compound with fluorine 18, iodine 125, and iodine 131, the importance of which I'm going to talk about a little later on. And what we did using isolated enzyme is showed that in comparison to this gold standard inhibitor, like I mentioned, this is DEAB, which has relatively high nanomolar binding affinity to aldehyde dehydrogenase 101, we can actually improve that again by 1 and even 2 orders of magnitude using this inhibitor. So effectively, this is the concentration of the inhibitor used. On the y-axis, it's the enzymatic activity of an isolated aldehyde dehydrogenase 101. The further to the left we get, the better the inhibition of its activity. So we can see with our fluorinated derivative and with our iodinated derivative, we actually improve upon this gold standard. Okay, so we have potential diagnostics and maybe even therapeutics. What we did is we then took a non-small cell lung cancer cell line that had very high levels of aldehyde dehydrogenase expression. This is A549s, and we knocked out ALDH1A1, as shown by this Western blot. And this is what happens with the fluorinated derivative. We get extremely high uptake in the wild type ALDH1A1 expressing cells. When we knock out ALDH1A1, we basically bring it back down to baseline. With our better inhibitor, the one with the iodine, we actually see a linear increase in the wild type A549 cells, going up to almost 300% of our administered activity per mg protein, which for those that are familiar with this is extremely high. FDG is probably around, I don't know, 10%. So again, an incredibly good binder. And then if we knock out ALDH1A1, we basically bring it down to the ground. We also are very fortunate in the fact that we have a facility that allows us to do some crazy things, which I'm really grateful for. And within our facility here, we actually bought a cell sorter that could handle radioactive compounds. And so we used this and asked a very simple question, is that if we can sort cells on their levels of aldehyde dehydrogenase activity, do we get the same differential in terms of the amount of radioactivity that's present inside these cells? So what we did is we co-incubated some ALDH high cells, both with our fluorine-18 diagnostic agent, NTX10, and also a fluorescent substrate that is trapped in high ALDH expressing cells. And then we could go and sort these cells to those that have high fluorescence, so high aldehyde dehydrogenase levels, and those that have low aldehyde dehydrogenase levels. And this is what we get with the scatter plot. We have some cells within this heterogeneous population that have high levels of aldehyde dehydrogenase, which is shown on the x-axis, and some that are also low. And because they're sorted, we can then go and put them in the gamma counter and ask the simple question is, where does our radiotracer go? Is it high in both populations, or is it high only in the aldehyde dehydrogenase positive population? And thankfully, we saw a very good differential here. So further evidence that our radiotracer is looking at ALDH positive cells. We've, of course, taken this in vivo. This is the same A549 tumor grown as a xenograft in this instance, and we see it lights up beautifully. We do get with our fluorinated tracer some liver uptake. We would expect that because ALDH1A1 is expressed in the liver, and it's hepatobiliary excreted. We also get a little bit of urinary excretion as well. When we grow the same tumor up, which is isogenic, it's only just got the ALDH1A1 knocked out. Again, you basically completely bring the signal down to zero. We can do another blocking study where we use a separate inhibitor, NCT501. It's not as great an inhibitor as our diagnostic agent, but it's pretty good. And even at high levels, we can pretty much bring that down to zero. Of course, this is PET. We can look at it dynamically, what happens in these tumors over time. We get increase in signal over time in ALDH wild type cells. When we knock out ALDH1A1, we get similar delivery initially, and then it washes out. And again, we get a nice blocking. So we're getting at least 3 or 4-fold differential between those that have ALDH low and those that have ALDH high. The tumor, we can look at whether it's metabolized, whether it's stable inside the blood and also in these various tissues. In the tumor, nicely, we see that it's not degraded at all, our diagnostic. We get some degradation in the liver, and that's what leads to this metabolite being formed that is then excreted into the urine. But again, in the blood, it's extremely stable. And actually, I would say that some degradation in the liver and its urinary excretion actually speeds up this process of removing background radioactivity. So we get a good tumor to background signal. Okay, so the really important and fun thing is that we want to go and identify therapy-resistant cancer. Going back to exactly that same model that I described before, this therapy-sensitive tumors, we don't get any uptake because there's very low levels of aldehyde dehydrogenase. When these tumors are treated, again, with a taxane in this instance, and they regrow, they have far higher levels of aldehyde dehydrogenase to try and resist that therapy. They outgrow, and we can image them nicely with our diagnostic, our NTX10. And again, you get these wonderful tumor to muscle ratios in the resistant tumors versus the sensitive. Again, of course, we can look at the time versus radioactivity curve in the therapy resistant versus the therapy sensitive, and we get at least a threefold differential. Okay, so we think that we can now identify using either XCT or ALDH1A1 in certain circumstances therapy-resistant disease. Now, for a long time, we said that was sufficient. You know, we're imaging scientists, so we say, okay, over to you, clinicians. You do something with this. You know, maybe you stop the treatment, and so these patients with resistant disease don't receive, at the end of their life, toxic treatments that make them very sick. Or we would say maybe you would go and try another therapy to try and improve the survival in these patients. But we thought maybe we could exploit the very mechanisms that we're imaging for therapeutic purposes. And this is exactly what we did. So again, this is led by the amazing former postdoc of mine, Hannah Greenwood, who really drove the NERV2 project, with ably supported by Abby, a PhD student in my group, who's taken this further and has done some amazing work. Now, what they did is they worked with this small biotech in the US, Agilvax, who were developing an antibody drug conjugate at XCT. So if we go back to that NERV2 story, you might think, okay, those that have therapy-resistant disease that have high XCT, maybe we can exploit that for therapeutic purposes. And this XCT antibody binds to an exposed extracellular domain on XCT itself, which is shown here in blue. Okay, so we asked a very simple question. If we took those NERV2 mutated tumors, H460 non-small cell lung cancer, and grew these up in mice, what happened with various different treatments? So this is what happens if we just give it saline. They grow very quickly because they're highly aggressive. And before even 10 days after we start this vehicle treatment, we have to cull the animals due to the tumor volume being too large. If we give them cisplatin, give them 2 doses of actually very high levels of cisplatin, we get a little bit of growth suppression with these tumors, a bit improved survival. But again, it's a highly aggressive disease. They don't respond very well because they're therapy resistant. But if we give the ADC just the 3 times, we get sustained tumor growth suppression. And that's even when they're off treatment. So we were really encouraged by this and think that we've got something quite remarkable. And again, we can see what happens here in terms of with the survival. There's actually not a statistically significant difference between vehicle and cisplatin in terms of overall survival, but there's a massive improvement, again, when we have this XCT targeted ADC. So this is one example where we can exploit increased XCT expression, which we can actually pick up using imaging and exploit it for treatment. Okay, but look, one of the hot topics within nuclear medicine is theranostics. It's been around for decades, many decades, but you know, there's a lot of interest now in terms of biotech startups and pharmaceutical acquisitions, given the success of PSMA. I sort of gave a bit of a strong hint before that this is, of course, exactly what we're looking at here. We've tagged our diagnostic and switched out the fluorine for imaging with an iodine. Actually, the iodinated version performs actually even better than the fluorinated version. And we think that we might have a small molecule binder that could actually specifically target therapy-resistant disease, which we think is quite unique. Of course, this is known as theranostics, so being able to treat the disease and use a companion diagnostic to select which patients we would like to treat. And we've actually spun out a company from King's just recently. These are the amazing co-founders for Nuclide Therapeutics. And we're really excited about where we could take this next into patients. But let me show you a little bit of preclinical data. If you remember with the diagnostic, we had some hepatobiliary excretion, which might be problematic in terms of the imaging agent. However, for the therapy, it's actually not too much of a problem. This is a time course biodistribution. So looking at where that radioactivity distributes over various different time courses, you can see we went up to 168 hours here in mice. But I think the really important one to look at here is the orange color, which is basically saying at 24 hours, where is the radioactivity? And you can't really see many orange bars here in this graph, right? You can see a little bit in the liver, a small bit in the large intestine, but pretty much by 24 hours, everything is cleared out from the animals. We get a little bit of deiodination, as you would always expect with iodine-based agents, radionuclides. This is shown in the thyroid, so we get about 15% injected dose per gram in the thyroid, but we can almost completely block that by giving potassium iodide, IP, every day to these same animals. Okay, we can also go and look at the dosimetry, and we get a really beautiful profile here. So I mentioned that with just 24 hours, it washes out really quite quickly. We see wonderful tumor uptake in those that have high ALDH expression. That's maintained up to 168 hours. So that therapeutic window for this radionuclide therapy is massive. And if we knock out ALDH 1A1, we don't get any tumor uptake. And again, this is just looking at tumor retention, where we have high ALDH 1A1 expression. We have a retention of our small molecule theranostic of nearly 5 days, which I think, again, is pretty unheard of. And that's because it covalently binds to ALDH 1A1. We've also done the therapy study here. So this, again, is a highly aggressive resistant tumor, grows rapidly when we have vehicle treatment. This is what happens with cisplatin. It's less effective even than the tumor that I showed you before. This is A549 tumors, but this is what happens if we just give it one dose of 50 megabec of our theranostic. Again, complete tumor stasis. And we have 100% survival of all of our animals that were enrolled on this study, whereas cisplatin doesn't do anything in terms of survival rates compared to vehicle controls. Okay, so I've told you a range of different stories, quite complex. I'm super excited to actually talk to you for the first time about all of these things. We're really focusing a lot in my group at trying to use the redox, tumor redox microenvironment and measures of oxidative stress to be able to pick up early response to treatment. We are now developing diagnostics to be able to even predict whether we're going to get resistant disease. And we think we can exploit those therapeutically in order to try and actually make a real difference in patients and target the very things that stop those tumors responding to standard chemotherapy and radiotherapy. So with that, there's a huge number of people I'd like to thank, past and former postdocs and students. One person I didn't really highlight, but has really driven a lot of this work is my exceptional radiochemist postdoc, Rich Edwards, who's sort of highlighted here. But again, I'm very fortunate enough to have an amazing team, lucky enough to have some extensive funding to support us to ask some of these crazy questions. Our support network within King's is exceptional. I've got great collaborators. And with that, I'd just like to thank you all very much for your time and attention.